Hello, I'm Dr. Israel Barkin, the Medical Director of the Prostate Cancer Research and Education Foundation. On our video blog, Ask Dr. Barkin, we talked in the past about Prostvac, which is a very promising vaccination, active vaccination, enabling T cells to attack the cancer. Recently, at the ASCO meeting, there was a presentation talking about a different approach, combining immunotherapy not only with other treatment modalities, but perhaps combining two types of immunotherapy with different mechanism of action, kind of giving the effect of punch 1-2, which means the first treatment is activating the T cells, and the second treatment enables, through the checkpoint inhibition, trying to have the T cells have the capacity to go after the cancer. Let's go now and look at the presentation here on ASCO meeting, and we see here intensified immunotherapy encouraging in chemotherapy-naive metastatic prostate cancer. And this is important because patients always welcome any treatment that avoids being on chemotherapy, especially if it's an immune therapy that doesn't have bad side effects. So they talked in this presentation about the Prostvac, which for those of you who want to learn more about Prostvac, please check the other videos that talk about Prostvac, including showing the mechanism of action. This is an important work done by Dr. James Galley from NCI. But here, what they try to do, they try to combine it with Ipilimumab, and I pronounce it quite okay, and I'll dare pronounce it even again, Ipilimumab. It's Ipilimumab, or the name they gave it, Yervoy. It's easier perhaps to say Yervoy, but I do not know what the origin of Yervoy. So in any case, they combined in phase 1 study, they combined the Prostvac together with these checkpoint inhibitors, and I would like at this point to get you acquainted with checkpoints. So let's view now a video animation that was created by Genentech. Let's view now the video.
As a scientist, I specialize in cancer immunotherapy. My work, and that of my colleagues, is to figure out ways to stimulate the immune system to kill cancer. To understand this concept, let's talk a little more about our immune system and how it works. Taking a look inside our body, all of us are born with an immune system that acts like the security checkpoint at the airport. Our immune system is like a high-tech full-body scanner system that protects us from things that are harmful. In the immune system, T cells have receptors that scan cells to distinguish between normal cells and abnormal cells, like virus-infected cells. Once detected, abnormal cells are attacked and removed by T cells. T cells also identify these cells for future recognition. The same T cells also play an important role in protecting us from cancer. Cancer cells are normal cells that have mutated. The more mutations cancer cells have, the more easily they are detected. Until recently, we didn't understand why T cells were detecting these cancer cells but not destroying them. But on a closer look, we've made some revealing discoveries. We've learned that one of the ways that cancer cells get past the security system is with the help of a molecule called PD-L1 that is found on the surface of cancer cells. PD-L1 is almost like a disguise that allows cancer cells to remain undetected. PD-L1 basically hacks the T-cell scanning system by jamming the signal to parts of the T-cell system called PD-1 and B7-1. Think of PD-1 as a sensor that's part of the T-cell scanning system, and B7-1 is like an alarm that issues an alert when something is wrong. When PD-L1 hacks the system, it prevents it from doing what it's supposed to do. In other words, it may prevent T cells from destroying cancer cells. Scientists have been working on fixing this problem and have designed molecules to target the PD-L1/PD-1 pathway. You can think of cancer immunotherapy as kind of like a software update for your immune system. Early studies suggest that by targeting the PD-L1/PD-1 pathway, T cells may be able to detect and destroy cancer cells. Let me show you in more detail. T cells have receptors that allow them to recognize cancer. Cancer cells can prevent elimination by turning on PD-L1. PD-L1 deactivates T cells by binding to PD-1 and B7-1. Blocking PD-L1 from binding to PD-1 and B7-1 could prevent PD-L1 from communicating with them. When this happens, T cells may finally be able to respond and kill cancer cells. Clinical trials are currently underway to understand the effects of trying to block the PD-L1/PD-1 pathway in the body. We are committed to studying cancer immunotherapy so that we can continue to find new ways to harness the immune system to fight multiple types of cancer. Brought to you by Genentech, a member of the Roche Group.
I would like to finish this presentation by first giving credit to the presenter at the ASCO meeting. This is Dr. Hapreet Singh, and also to give credit to ASCO. ASCO stands for the American Society of Clinical Oncology. If you have prostate cancer and you have an oncologist, I'm sure your oncologist has access to this publication, the ASCO Post, and there is even the ASCO Post iPad application that you could download it for you. But in any case, if you have any question about immunology, or specifically about pending studies in the field of immunology and prostate cancer, you could call our foundation, the Prostate Cancer Research and Education Foundation, at 619-906-4700, or write to info@pcref.org. Stay well, stay informed, and have fun. Goodbye.